16,000 men, hundreds of populations from the Atlantic to the Pacific, and one haplogroup that throughout the 20th century was referred to as Aryan. R1a is the golden thread woven into the Y chromosome, a genetic autograph that ideologists turned into a weapon. But when a team of geneticists led by Peter Underhill constructed a complete phylogenetic tree of this lineage, the results shocked the scientific world. The oldest branches of R1a were found not in the steppes or the Scandinavian fjords. 18 out of 24 carriers of the ancient form R1a-M420 turned out to be Iranians. The cradle of the Aryan marker is the gorges of the Iranian plateau. The ghost they sought in the north was hiding in the south. Who are the Aryans really? What does the blood of tens of millions of Iranians hold? The next 20 minutes will rewrite your picture of the ancient world. The word Aryan is one of the most poisoned words of the 20th century. It was appropriated by ideologists, smeared with propaganda, and soaked in the blood of millions. But long before swastikas and racial tables, it was simply a self-designation. Arya in ancient Iranian, Arya in Sanskrit. One's own, noble. This is how nomads descending from the Eurasian plains to the Iranian plateau referred to themselves. Not a race, not skin color, language and culture. Today, molecular genetics allows us to look beneath the layer of myths. 938 men from 15 ethnic groups in Iran provided blood for the largest study of the Y chromosome in the region. 84 markers, 10 microsatellites, and a genetic map that takes your breath away. Ahead lies a journey of 15,000 years, from the fires in the Zagros caves to the faces of modern Persians, Kurds, Lurs, and Azerbaijanis. This story will change everything you knew about yourself. Close your eyes and transport yourself 12,000 years back. The glaciers are still squeezing Europe in their cold grip, while fires are already burning on the Iranian plateau. The mountain village of Ganj Dare, western Zagros. Here, at an altitude of over 1,000 meters, live people the world will forget for millennia. They are just beginning to domesticate the first goats and learn agriculture. Geneticists call them Iran_N, Neolithic Iranians. Their DNA, extracted from ancient bones, became a key discovery in the large-scale project of Narasimhan and colleagues. These people carried a unique genetic component related to Caucasian hunter-gatherers, but distinct from all populations to the west and east. A living genetic island in the midst of the ancient world. And this component did not disappear. Iran_N remains the dominant foundation in the autosomes of modern Persians. The people from Ganj Dare did not go anywhere. They simply became you. But these people did not remain hunters. About 10,000 years ago, something happened at the foothills of the Zagros that forever changed history. The Neolithic Revolution. Wild wheat was sown into the first furrows. Barley and lentils filled the storages. Villages grew into towns, and towns into proto-cities. And a new genetic landscape was born. The Y haplogroup J2a, a silent marker inscribed in the chromosomes of men who farmed the first fields of the Fertile Crescent. Grugni's research showed J2a today constitutes between 22 and 25% of male lineages in Iran, and in certain populations of Zagros and southeastern Caspian, it exceeds 30. 10,000 years, and the marker stands firm as a mountain range. Every fourth or fifth man in modern Iran carries a direct paternal line from those first farmers. The grain sown in the ground of Zagros did not sprout only wheat. It sprouted a people. But J2a was not the only voice in this ancient chorus. Other male lines were emerging alongside it. G2a is a haplogroup found today from the Caucasus to the Pyrenees. Among modern Iranians, it comprises between 9 to 12%, while among the Arabs of Khuzestan it reaches 19. Another line, R1b-L23, is an ancient subclade that predates forms of R1b, which later flooded Western Europe. Among Assyrians and Armenians in Iran, its share soars to 24 to 29%. The Iranian plateau was not a genetic dead end. It was a reservoir from which ancient lineages spread westward, northward, and eastward. Waves of farmers carrying G2A and R1B moved through Anatolia and the Caucasus, merging into the genetic foundation of Neolithic populations in the Balkans and the Mediterranean. Imagine a map. At the center lies the Iranian plateau, pulsating and alive. From it, like arteries from a heart, flows a multitude of people and genes. Why can two peoples living in the same country be genetically as distant as different continents? The answer is geography. The Iranian plateau is a labyrinth. The Zagros mountain ranges form a wall that cuts off the west from the east. The deserts of Dashti Lut and Dashti Kavir slice the country in half like a heated belt. Each valley is a trap. Each pass is a filter. In these natural cells operated the master sculptor of evolution, genetic drift. Small, isolated populations lost some gene variants and accidentally consolidated others. This is why Grugni's research showed such a striking contrast. The Persians of Fars, living at the intersection of trade routes, accumulated the maximum Y-chromosome diversity. Meanwhile, the Arabs of Khuzestan and the Turkmen of Golistan found themselves at the opposite pole. The overall Y-chromosome diversification of Iranians stands at 0.952 thousandths. Mountains separated people, but the country hoarded diversity like a safe hoards gold. It was in this labyrinth of mountains and deserts that one of the most mysterious mutations in history was born. The haplogroup R1A-M420, the very marker we mentioned at the beginning. Isolation of a small group of men on the Iranian plateau, a random mutation in the Y chromosome of one of them, and a new lineage was established in their descendants. Quietly, unnoticed, without great battles. For millennia, R1A remained a local, rare branch. But about 5,800 years ago, a sub-branch R1A-M417 emerged within it, exploding into a demographic expansion of unprecedented scale. M417 split into two great branches, Z282, which moved into Europe, and Z93, which surged into the steppes of Central Asia, Afghanistan, and India. It is Z93 that will become the genetic signature of Indo-Iranian peoples. Think about this. A marker that originated in the silence of the Iranian mountains returned to the same plateau millennia later. From the endless steppes, riding in chariots, armed with a new language and new gods. But R1A is by no means the only witness to great migrations. There is a lineage that is much rarer and more mysterious. Haplogroup G1-M285. In the overall mass of Iranian Y chromosomes, it accounts for a modest 2 to 5%. It's easy to overlook, but it is within this quiet lineage that one of the most unexpected stories of the ancient world is hidden. Balanovsky's team conducted a deep analysis of G1 and found that the maximum diversity of its haplotypes is concentrated in western Iran. From there, it smoothly tapers off to the north, all the way to Kazakhstan. And here, the story takes a turn. In some Kazakh and Bashkir clans, the frequency of G1 soars to 80%. The answer lies in the founder effect. A small group of Iranian-speaking men migrated from the plateau to the north and produced a disproportionately large offspring. We tend to think that the steppe conquered Iran, but G1 tells a different story. Ancient Iranian-speaking tribes moved from Iran to the steppe. 4,000 years ago, the world cracked in half. From the north, from the endless steppes between the Urals and the Caspian, a wave moved that would rewrite the genetic map of half of Eurasia. The Sintashta and Andronovo herders, people of bronze, chariots, and fiery rituals. In their Y chromosomes pulsated the branch R1A-Z93, the Asian daughter of the ancient mutation M417. It became the banner of expansion. These people carried technology that changed warfare, light chariots with two-spoked wheels. They spoke a language that would split into Sanskrit, Avestan, and dozens of Indo-Iranian dialects. Narasimhan's genomic research showed that the steppe component of these herders was based on the Yamnaya culture with the addition of European farmers, and thus they reached the southern edge of the steppe world. Ahead lay the oases of Bactria, populated by descendants of Iranian farmers. The steppe met the plateau. Bronze met clay. The great synthesis began. At the intersection of steppe and oases lay a civilization that most people have never heard of. The Bactrian-Margiana Archaeological Complex, BMAC. Cities with massive walls, bronze seals, irrigation canals. The population of these oases genetically descended from the Neolithic farmers of the Iranian plateau, the component Iran_N, rooted in the caves of Ganj Dareh. And it was into these cities that the steppe people came, not with one blow. Slowly, generation by generation, the Andronovo herders seeped into the oases, trading, intermarrying, settling down. Research by Guarino-Vignon and Narasimhan showed the steppe and ancient Iranian agricultural components mixed in proportions ranging from 40 to 60%, complementing each other almost equally. This was not a conquest, it was a fusion. In the crucible of BMAC, a new genetic profile was born, neither purely steppe nor purely agricultural. A third, previously unseen one. The Bronze Age shifted to the Iron Age, and in Turkmenistan, a population emerged that geneticists refer to as Turkmenistan_IA. Behind this code lies a ready formula, descendants of BMAC, already imbued with steppe blood, already speaking early Indo-Iranian dialects. Not steppe nomads, not farmers, a new people with double memory in every cell. And this formula proved incredibly resilient. Guarino-Vignon's research showed a direct line of inheritance over 3,000 years. Modern Yugnobes, a tiny people in the mountains of Tajikistan speaking a language close to ancient Sogdian, inherited from the Iron Age population between 88 and 93% of their genome. Tajiks showed about 75%. 3,000 years of wars, empires, and invasions, and the genetic backbone stood firm. Like a code carved in stone, impervious to erasure. But if in Central Asia, steppe blood and agricultural foundation intertwined almost equally, then in Iran itself, the story unfolded differently. Steppe herders reached here too, but they did not drown it, but merely touched the surface. The ancient Near Eastern layer constructed by J2A, G2A, and millennia of local continuity proved too powerful to be washed away by the bronze stream from the north. The numbers depict a clear gradient. Quintana-Murci's studies showed that in eastern Iran, the share of R1A reaches 31%. The steppe signal here sounds loud. But in the west, around Zagros, R1A falls to mere percentages. The wedge from Central Asia fades, not reaching the heart of the country. That is why to speak of a single Aryan Iran is to simplify absurdly. An Eastern and a Western Iranian can be genetically further apart from each other than either is from a foreign neighbor. The steppe trace is merely one of the layers in the ancient mosaic named Iran. The steppe herders were far from the last to inscribe their line in the DNA of Iran. Haplogroup G1, page 08, a marker rooted in southern Mesopotamia and Arabia. On average in Iran, less than 10%, but among the Arabs of Khuzestan, J1 soars to 30. The genetic echo of the Arab conquests of the 7th century and centuries of ties with Iraq. From the northwest came its own stream. Among Assyrians and Armenians in Iran, the share of R1B-L23 reaches 24 to 29%, a Caucasian corridor along which genes flowed in both directions for millennia. And E1B, making up 6 to 7%, connects Iran with the Levant and North Africa. The Iranian plateau welcomed all. Every empire, every caravan added new letters to the genetic text. The Arab South, the Levantine west, the Caucasian north, layered over an ancient foundation without erasing it, but making it richer. The most recent wave came from the distant east. About 1,000 years ago, Turkic and Mongolian tribes surged into Central Asia and onto the Iranian plateau. They brought with them a Baikal component, an East Asian trace from the steppes of Mongolia. Among modern Tajiks, this admixture accounts for about 17%. But in Iran itself, the Turkic-Mongolian storm left almost no trace. Ghassemi's study showed that East Asian mitochondrial lines are found only in rare cases. The storm brushed the eastern fringes, left a thin coating, and receded. Here lies an important lesson. Iranian-Azerbaijanis speak a Turkic language, yet their mitochondrial DNA is almost indistinguishable from Persian and Kurdish. They changed their language but preserved their blood. Turkification turned out to be linguistic rather than genetic. Enough digging into the past. Let's unfold the map of our days. The maternal line passed from mother to daughter. Ghassemi's study read the complete mitogenomes of 192 Iranians from 4 groups: Persians, Kurds, Lurs, Azerbaijanis. 15 main MT haplogroups, 129 subclades. A genetic library that mothers have passed on to their daughters for millennia. Haplogroup U, 22.4%. H, 20.3. J, 13. H, V, 11. T, 9.4. All of them, West Eurasian. While East Asian and African lines combined do not make up 3%. But the most astonishing thing is not the numbers. Persians, Kurds, Lurs, and Azerbaijanis form a single cloud on the principal component graph. 4 peoples, 4 languages, and 1 blood through the maternal line. We have unfolded the maternal portrait. Now, the paternal one. The Y chromosome presents a much more colorful picture. While on the maternal side, 4 peoples are almost indistinguishable, on the paternal side, each region resonates at its own frequency. J2A, 22 to 25%, the Neolithic heart. R1A, from 0 in the west to 25 in the east. G2A, 9 to 12%. R1B, among Armenians and Assyrians, up to 29, the Caucasian corridor. J1, the Arab South. E1B, Levantine Echo. G1, Q, L, H. Rare voices from Central and South Asia. Each letter is a separate chapter, each percentage a millennium of someone's history. The overall Y chromosome diversification stands at 0.952 thousandths. Iran is not a monoethnic fortress. It is a genetic orchestra, tuned over millennia of migrations. If in Iran itself the steppe trace is barely distinguishable on the maternal line, then in the Hindu Kush it sounds in full voice. Mountain valleys of northern Pakistan, where Dards and Iranians still today live, speaking archaic dialects preserved by height and isolation. Here, Rahman's team read the complete mitogenomes of 213 people. About 8.4% of the mitochondrial lines were subbranches of haplogroups of steppe origin: H2A1A3, J1B1A1F, U4A1F, and subbranches W3A1. Their coalescent age ranges from 5 to 2,000 years. A perfect match with the Bronze Age and the expansion of Indo-Iranians into South Asia. Steppe migrants carried genes not only through the male line. Women traveled with them. Mothers passed down mitochondrial memory of the steppe to daughters, hidden in the valleys of the Hindu Kush, as if in a cache sealed in ice and stone. We have walked through the paternal line. We have walked through the maternal one. But the fullest picture comes from an autosomal analysis, the entire genome at once, thousands of markers across all 22 pairs of autosomes. It distinguishes 3 main components: three colors into which nature mixed the Indo-Iranian peoples. The first is Iran_N, the ancient foundation, ascending from the hunter-gatherers and farmers of the Iranian plateau. It dominates among Persians. The second is the steppe MLBA, a legacy of the Andronovo and Sintashta herders. Among Yagnobs and Tajiks, it is blended with the first in nearly equal proportions. The third is the South Asian AASI, a component of hunter-gatherers from the Indostan. It is hardly noticeable in Iran, but it becomes powerful among the Indo-Aryans of India. Three colors, three epochs, three worlds. Each Indo-Iranian people carries them in different proportions. This spectrum is the true heritage of the Aryans, not purity, mixture. So who are the Aryans? We have traversed 15,000 years, from the Zagros caves to laboratories sequencing mitochondrial DNA. And here is the answer. There are no Aryans as a biological race. They never existed. The word Arya denoted belonging to a culture and language, not a set of genes. Indo-Iranians were not pure-blooded conquerors, but heirs of a great mixture. Beneath the skin of the modern Iranian lies a living palimpsest, a Zagros hunter by the fire, a Neolithic farmer sowing grain in a furrow, a steppe nomad driving a chariot through bronze dust, a Levantine trader, an Arab horseman, a Turkic warrior. Century after century, life after life, and no layer can be torn away without destroying the whole. Nations are stories we tell each other. Borders are lines drawn yesterday and erased tomorrow. But DNA remembers only the wanderings. You are their proof. That's it. We have read 15,000 years recorded in nucleotides. We've traveled from the Zagros fires to the Andronovo chariots, from the crucible of BMAC to the mountain caches of the Hindu Kush. We have seen how J2A preserves the memory of farmers, how R1A-Z93 carries the echoes of steppe expansion, how G1 tells of the forgotten exodus of Iranian speakers to the north. The Aryans are neither a race nor a myth. They are one of the chapters in the book that the DNA of every living person writes. But this book is not closed. Every year, new genomes add pages that we have yet to read. And now, a question for you. Have you done a DNA test? What haplogroups have been discovered in your blood? Write in the comments. Subscribe to the channel, like, share this video. Because the past is not what is behind, it is what is within.